Hello everyone, I hope everyone doing well. Today let's see about biofertilizer. Before we go into this topic, what we are going to learn through this video is definition about biofertilizer, types of biofertilizer, then how we inoculate them, then methods of inoculation. First we see about definition of biofertilizer. Biofertilizer are nothing but there are natural fertilizer that are microbial inoculants of bacteria, algae and fungi which we use in separately or in combination which may help biological nitrogen fixing for the benefits of plants. They help build up the soil microflora and thereby the soil health. Biofertilizer also include organic fertilizer, manure, etc. Use of biofertilizer is recommended for improving the soil fertility in organic farming. Then, types of biofertilizer. Nitrogen fixing biofertilizer. Nitrogen fixing bacteria work under two conditions symbiotically and as free living bacteria, non symbiotic. The symbiotic bacteria make an association with the cropland through forming nodules in their root. The free living bacteria do not form any association but live freely and fit atmospheric nitrogen. Rhizobium. Rhizobium lives in the root hairs of the legume by forming nodules. The name rhizobium was established by Frank in 1889. The gene has 7 distinct species based on cross inoculation group concept. More than 20 cross inoculations group has been established so far. A new classification has been established for rhizobium that is slow growing rhizobium known as bradyrhizobium and the other group is fast growing rhizobium called rhizobium. Still this classification is discretely not distinguishable because the bacteria of one group may infect to other group. This is called the principle of cross inoculation, which relies on the assumption that a legume within a particular infection group may be nodulated by another species of nodule forming bacteria. Methods of rhizobium inoculants. The seed treatment has been found to be the suitable method of rhizobium inoculant. Some adhesive is used to make proper contact between seeds and inoculants, that is bacteria. About 900 grams oil-based culture is sufficient to inoculate. 10% jaggery solution is used as stacker for rhizobium cells to seed. First, the solution is spread over the seeds and mixed to build up a thin coat over the seeds. After associating the proper coating of slurry over the seeds, the inoculate is sprinkled over the seed and the content is again mixed thoroughly. Then, the content is dried in a shed by spreading thin on a polythene sheet at least for overnight. This is a free living or non-symbiotic bacteria does not form nodules but make association by living in the rhizosphere. Azospirillum species establish an association with many particular with sea plants such as maize, sorghum, sugarcane, etc. It is the most common organism and can form associative symbiosis on the large variety of plants. Azospirillum is recognized as a dominant soil microbe. Acetobacter. Acetobacter is a heterotrophic free-living nitrogen-fixing bacteria present in alkaline and neutral soils. Apart from its ability to fix atmospheric nitrogen in soils, it can also synthesize growth-promoting substances like auxin, gibberellin, and also to extend some of the vitamins. Many strains of Acetobacter also exhibit fungicidal properties against certain species of fungus. Responses of Acetobacter has been seen in rice, maize, cotton, sugarcane, pearls, millet, vegetables and some plantation crops. Features of Acetobacter. Features of Acetobacter contributes to the moderate benefits. Acetobacter is heaviest breathing organism and requires a large amount of organic carbon for its growth. It's poor competitive for nutrition in soil. It can benefit crops by nitrogen fixing, releasing of growth promoting substance and fungicide substance. Acetobacter is less effective in soil with poor organic matter content. It improves seed germination and plant growth. Vesicular or vesicular mycorrhiza, that is VAM. The term mycorrhiza was taken from Greek language meaning fungus root. This term was coined by Frank in 1885 and indicated above the mycorrhiza is a mutualistic association between fungal mycelia and plant roots. VAM is an endotropic. It lives inside the mycorrhiza. WAM help in nutritive transfer mainly of phosphorus, zinc and sulfur. They also mobilize different nutrients like copper, potassium, aluminium, magnesium, iron, manganese from the soil to the plant roots. They possess vessel sac-like structure from storage of nutrition and arbosicular for funneling them into root system. Mechanism of action. The worm forms an association with the plant roots. It penetrates into the root cortex and spread around the roots of the plant. As the name indicates, they possess sac-like structure called vesicles which stores phosphorus as phospholipids. The other structure called herbicicle helps bringing the distant nutrition to the vesicles and root. Methods of biofertilizer inoculation. There are three methods: seed inoculation, soil inoculation, self inoculation. First, we see about seed inoculation. This is the most common practicing of applying biofertilizer. In this method, the biofertilizer are mixed with a 10% solution of jaggery. The slurry is then poured over the seeds, spread on the cemented floor, and mixed properly in the way then a thin layer is formed around the seed. The treated seeds should be dried in the shade overnight and they should be used. Generally, 750 grams of biofertilizer is required to treat the legume seeds for 1 hectare area. Soil application. This method is mostly used for fruit crops, sugarcane, and other crops where localized application is needed. At the time of planting of fruit trees, 20 gram of biofertilizer mixed with the composite is to be added in the ring of one slapping. Sometimes the biofertilizer are also broadcasted in the soil, but we may require 4 to 10 times for biofertilizer. Before forecasting, the inoculant should be incubated with the desired amount of well decomposed. Self inoculation. This method is suitable for applications of acetobacter. In this method, 50 liters of water is taken in a drum and 4 to 5 kg of acetobacter fertilizer is added and mixed properly. Planting material required for 1 acre of land are dipped in this mixture. Similarly, if we are treating the potato, then the tubers are dipped in the mixture and planting is done after drying the materials in the shade. Let's see how we cultivate and culture the biofertilizer. First, we take a test tube, that is stock culture, then we undergo broth. If we need in a large scale production, we culture in a broth in big flasks, then fermented for large scale production. Then we will sterilize it and we will check the broth quality and blended with carrier. Let's packing it and quality checking after it will be distributed thoroughly to the marketing. Then advantages. There are many advantages of using the biofertilizer. They form an important association with the soil microbes and help in nutrient supply. Fix atmospheric nitrogen, increase availability or uptake of nutrition through solubilization or increased absorption, stimulate plant growth through hormonal or antibiotic action or by decomposing organic waste. They are cheap, hence reduced cost of cultivation. Improve soil property and sustaining soil fertility lead to soil enrichment. Build up soil fertility in the long term. They are eco-friendly and pose no damage to the environment. Disadvantages. Specific to the plants, rhizobium species culture does not work very well in the high nutrient tolerance strains of soybean. The acceptability of biofertilizer have been rather low, chiefly because they do not produce quick and spectacular response. Required skill is preparation and application. Difficult to store. Thank you for everyone to listen this. I hope everyone getting the main function of biofertilizer through this video.